Today, we're going inside a cell, specifically inside a hair follicle stem cell. And we're going to understand exactly what goes wrong in baldness and exactly what PP405 does to fix it. This is a science video. There will be biochemistry, but I promise you, it is genuinely fascinating. Let's start with the basics of cellular energy metabolism, because this is where the whole story lives. Every cell in your body uses glucose as its primary fuel. When glucose enters a cell, it goes through glycolysis, a 10-step biochemical process in the cytoplasm that breaks it down into a molecule called pyruvate, producing a small amount of ATP and reducing NAD+ to NADH along the way. Now, pyruvate has two possible fates. Option 1, it gets transported into the mitochondria via the mitochondrial pyruvate carrier, a protein complex made of two subunits, MPC1 and MPC2. Once inside, it enters the Krebs cycle and then oxidative phosphorylation, generating large amounts of ATP very efficiently. This is aerobic metabolism, high yield, oxygen-dependent, and associated with a quiescent, resting cellular state. Option 2. Pyruvate stays in the cytoplasm and gets converted to lactate by an enzyme called lactate dehydrogenase, LDH. This process regenerates NAD+ from NADH, raising the NAD+ to NADH ratio. It produces far less ATP per glucose molecule, but it is fast. It doesn't require oxygen, and it generates lactate, which turns out to be far more than just a metabolic waste product. Here's where it gets really interesting. For most of the 20th century, high lactate production was associated with anaerobic metabolism, the burning sensation in your muscles when you're sprinting. It was thought to be a byproduct, a sign of metabolic stress. UCLA researchers publishing in Nature Cell Biology showed something different. Lactate is a signaling molecule, and in hair follicle stem cells specifically, the intracellular level of lactate is a key regulator of whether the cell stays dormant or activates to drive hair growth. Low lactate equals telogen phase. High lactate equals anagen phase. The hair cycle is, at its root, a metabolic phenomenon. In a healthy follicle transitioning from rest to growth, there is a dramatic metabolic shift, from mitochondrial oxidation to glycolysis. LDH activity surges, NAD+ to NADH ratios rise. Lactate accumulates, and that combination tells the stem cells, it is time to grow. In androgenetic alopecia, pattern baldness, this transition stops happening. The follicle gets locked in oxidative, mitochondrial metabolism. The MPC stays open. Pyruvate floods into the mitochondria. Lactate production remains low. The metabolic signal for activation never arrives. And the follicle just waits indefinitely. PP405 is a small molecule that binds to and inhibits both MPC1 and MPC2, the two subunits of the mitochondrial pyruvate carrier. When PP405 is present, pyruvate cannot enter the mitochondria at normal rates. The effect cascades. Pyruvate accumulates in the cytoplasm. LDH is upregulated to handle the excess, converting pyruvate to lactate. Lactate levels spike. NAD+ is regenerated from NADH, raising the NAD+ to NADH ratio. The intracellular biochemistry of the follicle stem cell now exactly mirrors the metabolic state of a healthy, actively growing follicle, and the stem cells respond accordingly. They exit dormancy. They proliferate. They initiate the telogen to anagen transition. Hair grows. PP405 doesn't instruct the cell to grow. It recreates the metabolic environment in which healthy cells naturally decide to grow. There is an important distinction there. It is restoring a biological state, not overriding one. This mechanism isn't just theoretical. It has been validated at every step of the clinical process. In ex vivo human tissue, real facelift skin, a single application of PP405 increased LDH activity in follicle stem cells within 24 hours. The biochemical response to MPC inhibition happened exactly as predicted. In the Phase 1 first-in-human trial, 7 days of topical application produced statistically significant increases in Ki-67, the cellular proliferation marker, directly confirming stem cell activation in live human scalps. In Phase 2A, the trial of 78 adults, 31% of men with advanced androgenetic alopecia showed more than 20% hair density increases at 8 weeks. 0% in placebo. The hair that grew was terminal hair, thick, pigmented, fully formed, growing in areas that had been bare. A mechanism discovered in the lab, confirmed in tissue, validated in humans, and now moving to Phase 3 in 2026. What I find most compelling about PP405 is not just that it works, it is that we understand why it works. The science is coherent, from first principles to clinical outcome. That is rarer than it sounds in drug development. Phase 3 will be the real test. Larger populations, longer time frames, more diverse hair types. But the foundation is solid. For anyone tracking the NAD+ in longevity space, or anyone who is simply bald and paying attention, PP405 deserves to be on your radar. Like and subscribe. I'll be covering Phase 3 updates as they come. Full written breakdown in the description. Visit unbedsidemanners.com for more stories.